Yeah, we are live now. You can start.
Okay. Warm greetings to respected dignitaries, organizers, delegates, and one and all assembled here for today's session. I am Era Gupta, a third-year MBBS student at Bangalore Medical College and Research Institute, who will be moderating today's session. Heartiest welcome to the 5th day of Med-Invade 2021. It gives me immense pleasure to share with you all that Squad Medicine and Research, in association with JAYPEE, our educational partner, proudly presents Continuing Medical Education Series with eminent authors at Med-Invade 2021 conference. We have commenced this series and it will be continued throughout next week, every day from 3 to 4 p.m. Indian Standard Time. Starting with today's session under the series, entitled as Advances in Molecular Biology for Medical Students by our honorable speaker for the day, Dr. D.M. Vasudevan, sir. Welcome, sir. Dr. Vasudevan, sir, has 50 years of teaching experience. He has been a professor of biochemistry for the past 30 years. He was also principal and dean of various medical colleges like Amrita College of Medicine in Kochi, Kerala, and Sikkim Manipal Institute of Medical Sciences in Gangtok, Sikkim, vice principal of Government Medical College in Thrissur, Kerala for many years. He pursued his MD in biochemistry from All India Institute of Medical Sciences, New Delhi, and did postdoctoral research work at the Swiss Institute for Cancer Research in Lausanne, Switzerland. He also received research experience at the University of Health Sciences in Bethesda, Maryland, USA, WHO Immunology Laboratory in Switzerland, and Chester Beatty Cancer Research Institute in London. He is a fellow of Royal College of Pathologists, London, fellow of National Academy of Medical Sciences, and a fellow of Association of Clinical Biochemists. He is the recipient of the prestigious B.C. Roy National Award for Eminent Medical Teacher, adjudged by the Medical Council of India, which he received from the then President of India in 1994. He is a member of various academic bodies of different universities, as well as a member of the editorial boards of various journals. He has published more than 250 research papers in both national and international journals and presented more than 150 papers in international conferences. 30 students have already taken PhD under his guidance. He was also the president of the Indian Association of Clinical Biochemists of India for the year 2010. Respected sir, it is our honor and privilege to have you in our midst today, and we are very keen and delighted to learn from you. Before handing it over to you, sir, just a quick note for all our delegates that you can type in your questions in the YouTube live chat box or the Zoom chat box, and they will be addressed during the end of the session. Also, a post-event link will be posted in the chat box and also in the description at the end of the session. Make sure you fill the same to get eligible for the conference certificate. Now I shall be handing it over to you, sir. Thank you.
Thank you, Era, for the nice introduction. Today's topic is advances in molecular biology, and my presentation will be something like this, that there will be for the first 20 minutes or something like that, I will be describing the general molecular biology, that is DNA, replication, transcription, translation, and gene expression, something like that. That is the basic things. And then we go to a higher level, that recombinant DNA technique, gene therapy, molecular diagnostics, etc., where the advances will be coming. And for those who are going for the MBBS, the biochemistry examination, I will tell that there is the molecular biology topic comes about 25% of the total topics with regard to the marks, and therefore there is a weightage of this molecular biology. Comparatively, it is less than 25% if you take the textbook number of pages, but at the same time the marks are more. and therefore there is a weightage in that respect. And therefore it is very important that students should remember some of these basic things. The DNA structure you are familiar with, and there is the important thing to remember is that there is the base pairing rule. That is, wherever there is A, that is adenine, the opposite side there will be the thymine, and similarly if there is a G, the opposite side there will be G. And therefore that is that base pairing rule that is important for the replication part. The replication means that it is the new DNA strand is produced, and that is the semi-consecutive replication. That means a new complementary strand is synthesized over the old template. That is something like that. You can see the right-hand side that there is the red color, that is the old template, and over which the new template is produced.
Excuse me,
and
sir.
New strand is made based on the base pair rule. The old strand acts as the template. And
Excuse me, sir. So sorry for the interruption, sir. Sir, your
Yeah.
PPT is not being shared. Your PPT, you have to screen share it once, sir. The PPT.
Oh, sorry.
It's not being shared, sir. Thank you, sir.
But രണ്ടെണ്ണം. ഒന്ന് മാത്രം. അതെ മറ്റേ ഇതിലാണ്. പോസ്റ്റ് അറ്റൻഡ്. ഇതാണ്. So share screen. Can you see now?
Oh, yes, sir. It's being shared. Just a minute.
Yes, sir. Now it was.
Yes, sir.
Sorry for the trouble. And then this will be done.
Sir, again it was unshared.
Okay. Can you see now? Can you hear?
So we are audible, sir, but the screen sharing has not happened.
I don't know how to see. I had already shared it.
Yes, sir. It did share and then it disconnected.
Oh, that there was some.
So if you could try sharing once again.
Okay.
Yes, sir. It's visible now.
Good. No, I hope it is okay.
Yes, sir. Perfect, sir.
This is the semi-conservative replication, and the old strand. This is the old strand, that is the parent strands partially unbound, and the old strand is there. Over that acts as the template, and over that a new strand is made. Of course, all the base pair rule is always applied. And the enzyme is the DNA polymerase. This is the original strand, and that there will be, this is the C is there, and over that the G will be coming, and then the new strand, see, this is G is there in the template. Therefore, for that the C has to come, and therefore new C is coming there, and the new polymerase, the polymerase action will be there. And then the next T comes like that. Now, please remember that there is the RNA primer is needed for the DNA synthesis. See, if you take the old DNA strand, over that, suppose we want to start from here, that initially a primase enzyme will act, and therefore an RNA primer is added there. And then over that RNA primer, the DNA polymerase will act now that the new DNA strand is produced. And then that DNA is produced. And then later on, this primer is removed and another DNA comes there, and that completed new strand is produced. Therefore, that particular point you have to remember that the RNA primer is needed for DNA synthesis. This is the replication fork, that there is during the replication that there will be the DNA is partially wound and the DNA uncoils, the RNA primer is there and new DNA is synthesized. And a little more detail is given. The DNA could not be replicated, this area that is called a telomere, the end region of the DNA that cannot be replicated, that area is called a telomere. And therefore, because of that, a particular enzyme, telomerase, will come there. And that this particular area, that the telomerase enzyme itself contains an RNA template inside the telomerase enzyme that acts as the primer. And from there, the RNA is produced by the telomerase, acts as the primer. And from there, the DNA polymerase completes the lagging strand. And the DNA replication fork is something like that. The DNA is partially unwound, and therefore it will see something like that. This is called the leading strand in the sense that this is the new strand is continuously produced here. You can remember that this continuous production is towards the fork of the DNA. The opposite side, that away from the fork, that this production is discontinuous production, and small pieces are produced. That is called Okazaki fragments. And finally, these fragments are combined together by the DNA ligase. Now we go for transcription. Transcription means that the mRNA is produced out of the DNA. What is the difference between DNA and RNA? You are all familiar with. What is this? Ribose is here in the RNA, whereas the DNA is DNA ribose. Most importantly, you have to remember that in the RNA it is uracil, whereas in the DNA it is thymine. Wherever there is thymine in the DNA, when the new RNA is synthesized, uracil will be incorporated at that stage. The transcription process is quite simple, that there is certain areas will be identified by the RNA polymerase, and then that attaches to the DNA, and that is this so-called initiation process. And then that RNA polymerase moves on, and the new mRNA is synthesized along with that. And when mRNA synthesis is completed, that there will be a termination. This termination process, or the transcription termination, when the DNA RNA polymerase is reached at that particular region, the rho factor identifies the stop signal. And from there onwards, the RNA polymerase cannot work up. And therefore, the RNA polymerase as well as the RNA, the newly synthesized mRNA, is removed. Whereas in the mammals, it is an RNA, a rho independent termination. And therefore, that is the minor difference between mammals and the bacteria. Then one thing more to remember, that when the primary RNA transcript is produced, the whole region of the DNA is transcribed. That means exons and introns are transcribed. Exons means those areas which are exhibited out, that will come as the proteins. And therefore, these are the exons. The yellow regions are the exons here. Whereas in between, these in-between areas are called introns, which are not required for the protein synthesis. And after that, this is the post-transcriptional modifications are there. That there is, of course, the adding of 5' cap and the poly tailing, etc. are there. Moreover, there will be RNA splicing and removal of introns, and only the exons are combined together, and that is exported from nucleus to cytoplasm. And now we go for translation. That is when the transcription means that from DNA to mRNA, and from mRNA to polypeptide chain, that is called a translation. It is called translation because the nucleic acid language is changed to polypeptide language, that is the amino acid language. And now when they transfer RNA is important for carrying the amino acids, and that is the transfer RNA. The transfer RNA has a 3' end, and that is the acceptor arm carrying the amino acid, that is in the 3' end. And then there is the opposite side is called the anticodon arm, where the anticodon will identify the codons in the mRNA. And the anticodon arm of tRNA recognizes the codon in the mRNA. mRNA, suppose this is GAG, that is the anticodon, this is CUC will be there. And therefore, that will identify the codon in the mRNA is identified by the tRNA. And over that, this particular tRNA will carry that specific amino acid. That is how the translation is taking place. And the anticodon has base sequences complementary to that codon of the mRNA. The genetic code, you are already familiar with the salient features of genetic code. That is, they are triplet codons, that is 3 consecutive things will remain as the codon. They are non-overlapping and non-punctuated. These are degenerate. Why it is called a degenerate? Because there are 61 codes for the 20 amino acids. And that is why it is called a degenerate. Of course, it is unambiguous. That is without any doubtful meaning. That is one amino acid, one code will be translated as one amino acid. There will be no confusion in that. And now the protein synthesizing machinery is called ribosomal RNA or rRNA. In the mammals, that is 40S ribosomal and 60S ribosomal units are combined together to form the full ribosomal unit. Whereas in the bacterial ribosomes are different from human ribosomes. These are the bacteria, they are 30S and 50S subunits. And that is why the antibiotics will inhibit only bacterial protein synthesis, but will spare the human cells. That is because of this difference. That is, the bacterial ribosomes and human ribosomes are different. The translation process can be studied by activation of amino acid, that is B, initiation, C, elongation, D, termination, and post-translational processing. Quickly, we can go that the activation of amino acid, that is the amino acid plus tRNA, specific tRNA to that amino acid. The enzyme is amino acid tRNA synthase, and where each amino acid will have different syntheses, and amino acyl tRNA is produced, that the tRNA will now carry that particular specific amino acid, and this needs ATP. ATP is completely hydrolyzed to AMP level. Please remember, therefore, AMP level, that means that the compounds are therefore. Then initiation process, that there will be this. This is the pre-initiation complex, whereas the 60 S unit, and that carries with the methionine tRNA. Usually, the biosynthesis methionine tRNA is the first amino. Methionine is the first amino acid that is added, and then of course the methionine tRNA, JAYPEE, and certain elongation factors, as well as the 540 ribosomes with certain elongation factors are combined together to form this pre-initiation complex. Then some more factors and one ATP is used, and then there will be the initiation complex. Full initiation complex is produced, and here that methionine RNA in further 60S unit is also added to become this full unit. And here the 60S unit will have two areas. One is called A and P. That is, A is the amino acyl area, and P is the peptidyl unit, that site. And these two sites are there that they will be interchanged in the next elongation period. That is, the methionine RNA is there, and the next second amino acid is coming here with its tRNA. And then in the next step, these methionine and the threonine will be combined together. Of course, threonine is maybe any other amino acid here, and that the amino acids are combined together by peptide linkage, and then the dipeptide is now added on this second unit, and then this first tRNA is removed, and therefore the second amino acid, second tRNA will remain there. Now this A site is free, and to that the third amino acid, the tRNA containing the third amino acid will be combined, and this is the elongation process is repeated. Finally, in the region, but the termination sequences are there, and that termination sequence is identified. That there will be the amino acid, that the new amino acid is not coming. There is no specific amino acid for this UAA. Therefore, there is no tRNA is coming here, and therefore the polypeptide synthesis is stopped here, and therefore the free 60 RNA and the free 40S RNA, and then free tRNA, the free mRNA, and the termination factor, everything is coming out. Of course, that also needs JAYPEE hydrolysis, and the free protein is produced. The post-translational processing, the one important thing is proteolytic cleavage. Example is pro-insulin to insulin. This very nicely studied, and you have to study that pro-insulin to insulin, that post-translational processing, an important point. Then modifications of amino acids are there. For example, gamma carboxylation of glutamic acid of prothrombin. It is a vitamin K dependent process. Then hydroxylation of proline and lysine in collagen, that is vitamin C dependent process. There are also many other processes such as subunit aggregation, such as in hemoglobin and immunoglobulin. Protein folding, the three-dimensional structure should be correctly folded, and that needs chaperones. And then how this protein synthesis is a regulation, and that is mainly by operon. And the operon concept, that is the gene regulation. The lac operon has been identified very early. And the lactose metabolism is regulated by induction or de-repression process. Of course, this is taking place mainly in bacteria. It is worked out mainly in bacteria. Let us say in a bacterial colony that glucose is present and lactose is absent. In that case, the operon is shut off. And therefore, what happens that the RNA-P, that is RNA polymerase, will start the working here, but it will remain. When it reaches here, the repressor molecule is there. Therefore, it cannot move further, and therefore the structural genes are not transcribed. Whereas glucose is absent and lactose is present, then some small molecule, a few number of molecules or lactose are passing through the membrane, and that will reach the DNA area, and that will these act as the inducer. That lactose molecules act as the inducer, and that inducer will with the repressor. Now the repressor molecule it acts, and therefore the repressor cannot identify here, cannot bind here, and therefore the RNA polymerase is now free to action, and therefore that will move and on, and the structural genes are now transcribed. Therefore, that means when lactose is available, the lactose metabolizing enzymes are produced by the bacteria. That is because of this gene regulation, lac operon. In the human intestine also, lactase is an inducible enzyme. That is, you give lactose and then this enzyme will be induced. And it has clinical importance also, because after the age of 60 or something like that, there will be some persons, most of the persons will have this lactase enzyme will be absent. This particular mechanism will be absent. And therefore, that there will be the milk cannot be taken by those persons. Certain examples of de-repression is also there. Induction of tryptophan pyrrolase, transaminases induced by glucocorticoids. ALS in this is induced by the glucose. The opposite is also true, that there is the regulation of genes by repression. See, for the best example is the ALS synthase. The structural gene ALS synthase will produce the particular enzyme. That enzymes will produce the heme. When heme is plenty is available, that will act on the aporopressor. The aporopressor and the corepressor both together will attach on the operator, and then they promote the RNA-P. The RNA polymerase cannot pass through here, and therefore no more ALS synthesis is produced, and therefore no more heme. Therefore, it is an auto-regulatory mechanism. That is by repression. Now we go for higher topics, the recombinant DNA technology. And this is done by restriction endonucleases. These are the enzymes in order to transfer the gene. These are first to be selectively split from the parent DNA. And this is usually achieved by molecular scissors, technically termed as restriction endonucleases. These enzymes are from bacteria. In the bacteria, it is a natural activity that these enzymes will restrict the entry of phages, bacteriophages into host bacteria. And that is why it is called a restriction endonucleases. They are endonucleases that will remove the nucleic acids, and that will restrict the entry of the foreign DNA. That is their biological function in bacteria. But we are using it, we are isolating that and for our purposes. See, for example, that we have a human DNA here, and the bacterial DNA is here, and an echo R1, that is an enzyme that is used here so that it is selectively splitting particular area with having a specific DNA sequence is there. That particular area is spliced, is removed, and the DNA fragments that is now coming up will have the sticky ends, and these two sticky ends will come together, and therefore that this will become a recombinant DNA. That is the action of the restriction enzyme, in this particular case a CoR1. And now that can be used for admitting any human gene. See, for example, if you want to introduce insulin into bacteria, the human mRNA of insulin is taken and cDNA copy is produced, and to that cDNA is added to plasmid vector, and plasmid vector is cleaved by specific restriction endonuclease, and then that vector is now carrying the human gene, and the whole thing that the vector, the plasmid, is now entering into the, that is transfected into host bacteria, and therefore host bacteria will produce the insulin. Now selection of this bacteria carrying the human gene is by antibiotic sensitivity can be done, and that particular clone can be selected, and that particular clone can be amplified, and these those clones will produce the human gene proteins. That proteins isolation, that particular proteins can be isolated. Then human proteins, hundreds of human proteins are now being synthesized by this recombinant DNA technology. The best example is insulin. Insulin nowadays in market is now available. Human insulin that is produced by the bacteria. Of course, any number of such biological units such as interleukins, interferons, anti-hemophilic globulin. See, anti-hemophilic globulin is necessary for the if that is absent, that hemophilia will be produced, and the person will have bleeding tendency. And therefore, it is previously when we were students, what we were doing that is that the patient will get a blood transfusion, and this blood transfusion is then will be required every 3 months. That means that there will be overload of iron in those patients, and ultimately after a few years, after 10 or 15 years, the patient will have iron overload, and ultimately that is the end of the situation. Whereas nowadays, anti-hemophilic globulin can be specifically produced by the bacteria, and that is now available, and therefore there is no problem nowadays. Similarly, growth hormone, if you want to produce growth hormone, about 1,000 pineal glands from human persons, from human cadaver, are required to produce 1 unit of growth hormone. And so many units are required for the standard growth patients. And therefore, it is observed nowadays growth hormone is plentifully available because of the this particular technique. Hepatitis B surface, and any number of such examples are there. Now, cloning of animal, that is an advance that is taken place about 20-30 years back, that the nucleus is removed. Nucleus is removed from the egg donor, enucleation, and that this enucleated egg cell, the somatic cell is taken from a donor, and that donor cell is added, the nucleus is added here. Therefore, nucleus is coming from here, and the egg is coming from here. And now that is a zygote and blastocyst, and that is given to a foster mother. And from there, any number of such animals can be produced. That is called cloning. Now, the advantage is that same, suppose there is a specific trait here. Suppose this particular donor can have a specific tendency, let us say that will produce alpha AHG that is producing there. That tendency can be transferred here, and any number of such animals can be produced. That is the advantage of the cloning of the animal. Of course, Ian Wilmot cloned the sheep in 1996. A great the science has been come forward in a great step by the stem cells. Now this is the era of stem cell generation, and previously uncontrolled or untreated diseases can now be treated. The totipotent stem cells can be taken, and that is cultured. That will become pluripotent stem cells, from where you can identify, you can produce, specifically produce hematopoietic cells that will go to blood cell series, or neural cells that will go to nerve cells, or they can go to mesenchymal stem cells that will go to bone or cardiac cells, or something like that. Therefore, any number of cells, any number of specialized cells can be produced from totipotent stem cells. And these stem cells can be harvested from bone marrow. About 1% of bone marrow cells will have these stem cells. Or from peripheral blood, of course, the harvest, it will be very less. The umbilical cord blood is a very good area where you can get some such stem cells. Nowadays, stem cells, stem cell banks have come. Therefore, you can get such stem cells. And this is the, as I have told, that this is the in future, that newer and newer methods of treatment will be available by this method. This has just come in, and therefore this is the real advance in the science. The stem cells, there are so many clinical applications. Already there are a few clinical applications. See, for example, hematological malignancies. See, chronic myeloid leukemia, there is usually they will give the drugs, and after some time all the drugs will be resistant, and then there will be difficulty to the patient. Then sometimes that they will do, what they will do is that they will remove the whole of the hematopoietic system. That is, total body irradiation can be given, and the whole hematopoietic system will be removed, so that all the cancer cells will be removed, but at the same time normal cells are also removed. Patient cannot survive unless you give stem cells. The stem cells can be given, and that will go as the hematological cells, and from there the new cells can be generated, and therefore patient can survive. Similarly, secondary bone marrow failure syndromes, genetic disorders of hemoglobin, congenital immune deficiencies, inborn errors of metabolism, and many of these areas where stem cells have already been used for clinical applications. There are many other newer applications that is coming, say stroke, area of brain is gone. And what can you do? You can give such cells, and that will regenerate. Parkinsonism, coronary artery diseases, diabetes mellitus, Alzheimer's disease, severe burns, various types of cancers. These are all in the still in the experimental stages, but still work is going on. There are very good results are also coming out, and therefore in the future, maybe after 10 or 15 years, when you people go as the senior doctors, by that time, these things, stem cells, will be a very useful treatment modality. Gene therapy is already in the market, that here they take the cells from the patient. Of course, that particular gene is absent here. And these cells, the retrovirus carrying normal gene is introduced here. Therefore, the normal gene is introduced into the patient cell by the virus, and the patient cell now contains the normal gene. And the cell with the normal gene is then injected to the patient back. And therefore, the theory is that this particular cells will multiply, and therefore the gene will be there in the patient. And therefore, normal gene will be produced, normal protein will be produced, and therefore that particular disease will be eradicated in that particular patient. Now, what are the vectors? Vectors are viruses are generally used. Retroviruses, adenoviruses, and herpes simplex viruses are used. Non-virus systems, liposomes, plasmids, and retroviruses are commonly used. These are RNA viruses that replicate through a DNA intermediate. The Maloney-Murine leukemia virus is commonly used agent. Here, the retrovirus wild type RNA is taken, and from that the GAG, POL, and ENV genes are removed because that is necessary for the replication of the, and therefore that will be removed. In that place, add the cassette containing the human gene. The correct gene is now here. This is now a defective virus that is put into a packaging cells which will provide the viral code. If there is only the viral code, then only they can replicate into the human host. And these are now infective. That means that they can enter into human cells, but they cannot replicate to produce the disease. And there, this green is a human cell, and that is a receptor mediated entry of retroviruses into the human host cell is there. And in the human cytoplasm, it is reaching there. And from the reverse transcriptase of the virus will copy RNA into DNA, and into the DNA that is integrated into host DNA inside the nucleus. That is the way by which we generally introduce the foreign gene into the human cells. There are many success stories. For example, severe combined immunodeficiency, where the adenosine deaminase deficiency is there. And this is happened in 1990. And Duchenne muscular dystrophy, the dystrophin, a particular gene is now absent in these particular patients. Cystic fibrosis, CFTR gene. Similarly, wherever there is a single gene is absent, that gene can be introduced theoretically. There are many success stories, but there are many stories are there where it is not taking place also. Therefore, it is still more or less in the experimental stage. But you can see that spinal muscular atrophy is a condition where the anterior horn cells of spinal cord atrophy, so muscle cells are degenerated. A particular gene that is called SMN1 gene, that is, it is named as survival motor neuron, and that is it is gene therapy by commercially now available. This is the commercial name of that particular the drug that is now commercially available. This is the gene therapy, classic gene therapy, and that is one shot, one injection, intravenous injection is supposed to be sufficient for correction of the spinal atrophy. Of course, this is also in the very early stages of clinical thing that there should be more and more work has to be done. But what I want to emphasize is that the gene therapy has already come in the market. And therefore, by the time you become practitioners, naturally there will be more and more gene therapies will be there. The molecular diagnostic is a very big chapter, in the sense that nowadays almost all diagnosis, the bacterial or viral, all these infectious diseases, as well as the genetic diseases, all these are now identified more or less by molecular biology techniques. The DNA hybridization is a this thing, that isolation of the DNA from sample, and then that is the denatured, that is the single stranded DNA is made. Into that, the labeled DNA probe is added. Probe means that the labeled DNA is added, specific DNA part. So suppose you want to identify whether the particular virus is there. The virus gene is labeled, and then that is DNA probe is added to the human gene, and therefore that probes will bind to the gene of interest, and thereby we can say that this particular gene, suppose that you want a gene of virus is to be identified. Here you can see that the virus gene is present in this DNA. This is the southern blot technique. That is, the DNA is made into fragments, and then that is electrophorized in the agarose gel, and that is transferred in the nitrocellulose membrane, and that is to that you add the labeled DNA probe, autoradiography, and your particular interest. Suppose this you have this particular gene is interested here. That particular gene is now you can identify by the autoradiograph. This is the same thing as southern blot, which I had already told that the restriction enzyme, gene electrophoresis, and the DNA probe is added, the result will be the positive or negative. That is, the southern blot is for DNA. Now, if you want to identify RNA, that is called a northern blot, more or less same, single-stranded, the RNA is there, and that agarose is passed through gel electrophoresis, and then single-stranded complementary DNA is added as the probe. Again, you will get the labeled probe that the specific mRNA is identified. If you want to identify protein, that is called a western blot. Here, the protein is isolated, and that is passed through acrylamide gel usually, and that is separated, and primary antibody to specific polypeptide is added, and then you can identify that particular protein. So for southern blot, northern blot, and western blot, for identification of DNA, RNA, and protein respectively. And as I have told, that the abnormal genes can be identified by the molecular mechanism. For example, sickle cell gene, that there is, and this is the normal gene, and one MST2, that particular restriction enzyme will cut the normal gene into two parts, whereas in the sickle cell gene it will cut only one area. So if you pass through that, and then autoradiogram southern blot is done, and then you can see that there is two bands in the normals, whereas there will only be one band in the sickle cell patient. So for that you can identify like that. Polymerase chain reaction, it is very commonly done in almost all molecular biology lab. That is the standard basic thing. That is, the original DNA is taken, denatured at 95 degrees centigrade. DNA primer is added and annealed at 50 degrees centigrade. And then there will be extension by the DNA that by Taq polymerase, and that at 72 degrees. So that is why it is called chain reaction. That is 95 degree, 50 degree, 72 degree. This is every 2 minutes that there will be the temperature is altered, and that is why we call it as chain reaction. And polymerase chain reaction. Polymerase is the enzyme. The Taq polymerase is the enzyme. And this step 1, 2, and 3 are repeated many times. The first cycle you will get 4 strands. Second cycle 8 strands. Third cycle 16 strands. If you make it about 30 cycles, there will be 30 million copies are produced, and therefore large quantity of DNA can be synthesized. You take one bacteria, very small number, only a single bacteria you have, and then that you multiply 30 billion times, and then you can do any number of tests. That the test can be done. Just you can identify that only when you multiply the DNA. That is the importance of the polymerase chain reaction. This is used for diagnosis of bacterial and viral diseases, medical legal cases, diagnosis of genetic diseases, prenatal diagnosis of inherited diseases. The list will go on like that. The crime detection, you can see that this is the S is the suspect number 1, suspect number 2. And this is the evidence. Evidence means that there will be one hair follicle is taken at the site of the crime. You can see that this S2 and E that will very correctly match, whereas S1 will not, the strand will not match that. Similarly, you can put the paternity cases. See, for example, this is the sequence. This is the DNA pattern from mom. This is from dad, and this is 1, 2, and 2 or 3 other children. You can see that the mother and these patterns. This will never. These particular green things are not here anywhere, and therefore you can immediately say that this son is adopted. Similarly, we can identify the paternity, and that is what we generally call as DNA fingerprinting. That blood sample is taken, DNA extracted, DNA is cut by restriction enzyme, and the DNA fragments are separated by electrophoresis. Southern blotting is done, and this DNA probe is hybridized, washed, and radioactivity, then X-ray film is added, film developed, and then you will get a particular pattern. And this particular pattern you can identify. That is what we call as DNA fingerprinting. Each person will have specific pattern, just like fingerprint in your thumb. Similarly, each person will have that DNA pattern will be different. The a modification of the PCR is the reverse transcriptase PCR, RT-PCR. And instead of tag polymerase here, they teach the polymerase. Therefore, thermophilus is added. And that is, so mRNA, it can be copied to cDNA synthesis. Therefore, here from the RNA to DNA, and then that DNA can be, in the ordinary PCR, DNA is detected. That DNA could form a living or non-living organism. Here, the reverse PCR, mRNA is detected. That means that it is derived from a living organism. That is the importance of reverse transcriptase reaction. Not only that, RNA viruses can be identified by reverse transcriptase reaction. Now there is the real-time PCR. Real-time PCR, you can quantitate the DNA that is present. See, for example, originally you do 0.01 nanogram, here 0.1 nanogram, 1 nanogram, 10 nanogram, 100 nanogram, like that originally you put, and do the PCR reaction, and ultimately you will get, the machine will give such a picture. And now your test tube will contain the unknown quantity. That will also be same test is done. And suppose that comes like this, then immediately you can say that the unknown sample contains 1 nanogram. This quantitation of the DNA is absolutely important in certain diseases. See, for example, in clinical practice, viral load of HIV, that is human immunodeficiency virus, or HBV, that is hepatitis B virus. These two are important for clinical situations where the treatment, the dosage of the drugs are determined by the number of particles, that is viral particles, and therefore this real-time PCR becomes absolutely important in such situations. Still further modification, very recently come modification is the LAMP technique, loop-mediated isothermal amplification. That is that the DNA, that there are certain primers are added, and then ultimately that there is a loop-like formation is taken place, and then from there the exponential amplification is taken place. What is the advantage of loop-mediated LAMP technique out of the previous technique? The advantages is that the amplification of DNA takes place at an isothermal reaction. That is why that isothermal, that is only at a 65 degree. Whereas in the PCR, it is the 90 degree and then 50 degree and then 75 degree. It is cyclic reaction is there. Whereas here it is isothermal. That is all the time it is 65 degree. And therefore a simple hot water bath will be sufficient. The big machines are not required. It is a quick one-step and user-friendly rapid reaction. And there is no need for expensive thermocycling equipment, etc. And the final assay, the LAMP assay, takes less time for amplification and detection. These are the real advantages. The RT-PCR that you must have already heard about during this pandemic. The COVID pandemic, that usually the COVID virus is identified by RT-PCR. The alternate technique that is recently come is the LAMP technique. LAMP technique has certain real advantages. That is, these are isothermal, and it needs only 20 minutes, whereas the PCR needs 2 hours. In the RT-PCR, that is the usual commercial technique, that it will take about 10 hours to get to the result. Whereas here it will take only, the LAMP technique, it will take only 1 or 2 hours. and the yield is also high, and the detection cost is very low. And the DNA sequencing, just one or two sentences, we can finish that. DNA detection mutations is Sanger sequencing method is the Sanger sequencing technique. I am not going in greater detail, but that is it is given in our textbooks that you can identify the number, the actual sequence of the nucleotide in the DNA, and that is automated nowadays. The automatic sequence that there is the identification that there is two nucleotides are here added here. Mutation has taken place that can be identified by the automated sequencing. Previously, the human genome technique that originally when we started, that it took many years to complete the human genome sequencing, whereas in this automated sequencing nowadays available, it will take only a few hours. Therefore, that is the difference. The years and hours, that is the difference. Now, that is the advance that has taken place in this particular area. And further taken has played, the next generation sequencing has taken place. The NGS is used. The difference is that these are analyzed by millions of small fragments are taken by the DNA, and that is parallelly sequenced. Therefore, many total human genome will be made into many thousands of pieces, and each of them are taken together, and therefore quickly you can get the result. And it is used in clinical genetics, microbiology, oncology, etc. And another great advantage came as the microassay. Here, the DNA sample, the experiment is taken, and that is each blue spot here indicates the location of a PCR product. On real microarray, each spot is about 100 micrometer. And here the RNA is isolated, and from that a cDNA is produced, and that label, that usually fluorescent label is added. And in that, to the gene sequence, the genes are already put in this array, and therefore the hybridize, and you will get such a result that here gene is not active or gene active but normal, gene active but disease that is highly active. All these you will get immediately. That is, thousands of genes can be identified by within a few hours. That is, whether that particular gene is active, that particular gene is inactive, etc., can be identified. Please remember nowadays that it is very common now in advanced laboratories that hundreds of genes are combined are tested together, and then you will get the result immediately. This is a laser technique. This is a very commonly used technique nowadays, lateral flow technique. That is, actually there is one strip will be there. Of course, that will contain many layers. For example, in the first layer, that blood will be absorbed, and then that is the separation membrane is there. Then there is the adhesive tape, and that the PET, the upper layer is there, that particular the PET, and from that sample is then it is taken towards this adhesive tape is here, and therefore through that the water molecule is going out in the filter paper, and then this is the enzyme immobilized film, and where the reaction is taking place, and then unwanted liquid is absorbed by here, and that can be identified. And therefore, this has been nowadays. See, for example, you might have already heard that this is COVID situation. See, for example, the antibodies and antigens can be detected that by lateral capillary flow. See, for example, in this particular case, that here the specific antibodies are already attached on the plate, and to that you add the serum of a suspected person. Suppose that contains the COVID antigen. These are COVID antigen. And then that will fix there, and then that will react with the second antibody, and then you will get a color. Therefore, when there is a color, immediately you can say that there is a reaction taken place, and that means that the patient is positive. That patient has the antigen. Like that, there are any number of situations you can make. And this is the future, that the current medicine is that you have a lot of patients, but you have one therapy. Suppose one particular disease, one particular therapy only. And what will be the result? Most of them will be cured. Some of them have no effect. Some of them even have adverse effect. That is what is happening nowadays. We want to change it. And that is the future medicine is personalized medicine, where, let us say, these patients are there, but their DNA is taken, and this DNA is identified what particular treatment modality has to be given, is to be identified, and this group of patients will give this particular drug, and this group will have this particular drug, and then this blue person will have blue drug, and therefore after giving the therapy, all patients will have effect, and therefore there will be complete cure. Therefore, that is the future of medicine, and the molecular medicine is an important part to play. With that, I remain. Thank you very much for your listening. Thank you. Good.
Thank you so much, sir. This was truly an insightful presentation, integrating clinical aspects, especially focusing on the current COVID-19 pandemic and the recent advances in the field of molecular biology. And I'm sure the audience feels absolutely enlightened. We will now be taking questions from the audience. Please feel free to type in your questions in the chat box, the Zoom chat box or the YouTube live chat box.
If any one of the participants in Zoom would like to ask any question, they can unmute and ask. Sir, we have one question.
Yes.
Sir, like the role of molecular biology in Indian physicians, I mean, the usage of molecular biology in Indian physicians is very low. Like, what are the suggestions for the young doctors and inculcating the molecular biology in their daily practice?
As I have told you, that the future of medicine in molecular biology, that is, see, as I have some aspect I had already told. See, for example, the molecular medicine, the gene therapy has already come in market. Of course, it is now very costly, but at the same time, it has already come in market, and therefore, within few years, that will become a common practice. Similarly, the diagnosis part, that all the PCR techniques and all the molecular biology techniques. Just for example, the pandemic, that is the COVID, that diagnosis is dependent on RT-PCR and LAMP techniques. Therefore, this has come in the clinical aspect, complete and basic molecular biology knowledge is absolutely important for an ordinary practicing medical specialist.
Sir, one more question is, like, sir, what are the future opportunities of like pathways for medical students to get engaged into the molecular biology?
That is how they can acclimatize with that.
Yes, sir. Yes, sir. To take it as a career pathway.
Career pathway. Career pathway. Say one is that, of course, the MD biochemistry, and then the specialization in molecular biology, that can see any molecular biology laboratories. Nowadays, there are any number of molecular biology laboratories are coming in the nation, in the country. That and all of them will need an MD person, according to the to the our Government of India regulations that they need an MD biochemistry person or a pathology person to man the laboratory, clinical biology, biochemistry laboratory. Therefore, the biochemistry is a very advancing many new techniques are coming up, and therefore there will be the sky is the limit for such a persons entering into the biochemistry person. There will be no problem for a future. And of course, that some of them can really go for research work also. That newer techniques can be identified, newer drugs can be identified. This the pharmaceutical industry is another area where MD biochemistry and specialized biochemistry persons can go. Therefore, these are any number of areas where an MBBS person can go for biochemistry. I am an example.
Yes, sir. Sir, one more question is like how much aware the student should be about the latest advances in molecular biology or molecular genetics. Like recently there are many advances like stem cell therapy and optogenetics. Recently CRISPR-Cas also emerged.
Yes, yes.
But the information for the medical students is very limited in their curriculum. Like, what are your suggestions to keep updated about these experts, and what is the need for medical students to keep updated about these latest advances of experts?
Yes. See, for example, when I was a student, at that time, molecular biology was only a few pages only in the biochemistry textbook. Even protein biosynthesis, the technique was not known at that time. That I am telling about 50 years, 70 years back. Therefore, the advance in biochemistry is such a rapid pace that it is absolutely necessary for the person to keep in touch with that. Then only you can identify. See, for example, today's topic, my textbook will contain that there is only a small paragraph on CRISPR. But that technique, that the specific cutting of the DNA, that particular technique has come now in the clinical medicine. That is within another 5 years, that will become a part and parcel of genetic diseases, treatment of genetic diseases. Therefore, how it can be done is that only that particular person has to see, the person interested has to be familiar with the newer techniques, has to be studied. That is the only way. But that is absolutely important for even general practitioner should have some knowledge on these advances in biochemistry. That is why I brought some of these advances just to stimulate you that there are certain things coming up within another 10 years that will come to the clinical practice. That is why, that is the purpose of my discussion.
Excuse me, sir. You had
Yes.
mentioned about the LAMP technique. If you could enlighten us more on how it's clinically relevant, not only with respect to the current COVID-19 pandemic, but otherwise also, what is the future we have with the LAMP technique?
Theoretically, almost all PCR will be changed to LAMP in due course. May not be immediate, but within another 10 or 15 years. Because one, if you want to establish an RT-PCR laboratory, about 30 to 45 lakhs of rupees are required for the infrastructure. Whereas for the LAMP technique, you need an ordinary lab, perhaps 1 lakh or 2 lakhs will be sufficient. That is the difference. Therefore, any new lab that wanted to go for molecular biology technique will opt for the LAMP technique. The LAMP technique is a real wonder that within 1 or 2 hours you will get the result. Whereas RT-PCR you need 10 hours. The result will be available on the next day. Here the result will be available within 2 hours. That is the difference.
Right.
And not only the COVID, any DNA or RNA, virus or microorganism, whatever may be, that can be identified by this method. It is very versatile technique, and many of these kits are now coming up. The technique has come is only about 8 years back in the world market, and in India it has come only recently. That is why it is not very familiar for you people, but that is why I want to introduce that. It will come in the future. That is the technique that is coming up. And the present PCR technique, but the RT-PCR technique will be changed to LAMP in due course.
Right, sir. So there's one more question about gene therapy. Is it true that it can be done only in childhood, as in up to a certain age? And if yes, why?
No, it is not absolutely important in the childhood, because why we are doing that in the childhood? Because most of these major disease, major damaged persons will be children. See, usually these type of children will die within about 1 or 2 years. And if you give the correct treatment, they will live a useful life for a reasonably good life for a 50, 60 years like that. That is the importance of the genetic therapy. But at the moment, the present day, our knowledge with the present day, that only single genes can be transferred. Single gene, if they see, for example, phenylketonuria, or that these things can be treated well, theoretical, by this gene therapy. Because of, but many of the diseases, for example, diabetes mellitus, it is a multifactorial and multi-gene defect. In such situations, our theoretical knowledge has not yet come to reach that level. That is the only.
Yes, sir.
It will take some another 20, 30, 40 years like that. Certainly there further, but my emphasis is that future of medicine, future is by molecular techniques.
Yes, sir. So we have one more question from the audience. Is
Yes.
personalized treatment being followed anywhere in the world right now?
Oh, any number of cases. See, for example, most of the cancer cells. See, for some cancer cell will have some particular gene. See, for example, EGFR gene. That particular gene is mutated. That is why that cancer is produced. It's very common in gastrointestinal malignancies. And it is a routine thing that we are doing in almost all major molecular biology laboratories, where the EGFR test is done. If it is positive, there is a specific antibody, molecular monoclonal antibody available that you give, and then the disease will be, I mean, you cannot say cured, but almost cured. That is the 10, 15 years back, you cannot even think about that. Such. But if you take 100 persons, there may be only 20 persons for that. That is why I am telling personalized medicine. That is specifically given those persons where that particular gene is mutated. And in some other person, there will be some other gene will be mutated, and you will give specific antibodies against that. That is now come for many of the cancer therapy. Nowadays, it is monoclonal antibody, different types of monoclonal antibodies, and that is specific to that particular gene that has come. Yes.
Right. Yes, sir.
Yeah, proceed for vote of time.
Okay. With this, we have come to the end of the session. I would once again like to extend my deepest gratitude to Dr. Vasudevan, sir, for taking time out from his busy schedule to enlighten us all and to answer the queries put forth by the audience. This has truly been a great learning experience for us, sir. I would also like to thank our lovely audience for tuning in for the session. Hope to see you all in the consequent sessions, and make sure you fill the post-event link in the description to get eligible for the conference certificate. Thank you.